Good morning, everyone, and thank you for coming to our tutorial. My name is Atul Butte, and I'm the new director for the Institute for Computational Health Science in the University of California, San Francisco. I've been there for about a month, and prior to that, I've been at Stanford for 10 years. I'm also the principal investigator for ImmPort, a clinical trials dissemination system that is funded by the National Institute for Allergy and Infectious Disease. And you're going to see ImmPort represented by several speakers this morning. I wish you could be there in person, but the new duties here at UCSF are, to put it mildly, overwhelming. So there's no chance for me to make it across the country this weekend. So instead, I'm giving this introductory lecture virtually, and I thank you in advance for your patience as I go through this introduction. Now, what I really want to start with is this idea that we are in a new era for science, and that's specifically of this era of sharing data, sharing methods, sharing what we're doing in a raw sense. And I just want to start by just showing you how others share their data before we get to the clinical trial space. Some of you know that cancer researchers share their data. There's a flagship project that actually just completed called the Cancer Genome Atlas. This was funded for many years by the National Cancer Institute. And you can go to the TCGA website and see how this incredibly large consortia has studied more than 20 types of cancer, 11,000 subjects, and data on DNA mutations, RNA measurements, some protein measurements, methylation patterns, even some of the imaging, for example, brain imaging and mammography, are all publicly available through the TCGA portal. So this is an amazing example of how the NCI got a team, a massive team of cancer researchers to share their data with each other and with the public. You may know that genetics researchers share their data today. And here the mandate comes from the funding agency as well. This repository I'm showing you here is called dbGaP, the Database of Genotypes and Phenotypes. And while there are genotypes, the phenotypes are actually quite interesting. It's not just a genetics database. Here, the mandate to share comes from the funding agency, as I said. Specifically, if NIH is funding you to do genetics research, you must share your data into dbGaP, many times before publication. So actually, the data sharing mandate actually happens before the publication. But to be fair to researchers, we give them a chance to get their own papers out. And you can see here in the middle of the slide, there is an embargo date. So one is allowed to analyze this data, but it's not allowed to write your own paper until the original team has a chance to write their paper first. Why is it worth this kind of hassle? I just want to point out in tiny font at the top is actually the Framingham Heart Study. Think about an amazing 70-year longitudinal study. Framingham, of course, west of Boston, where that cardiovascular system has been studied by almost 2 generations of individuals. It's amazing to me still that one can download now the genotypes of 14,000 people, but perhaps even get 70 years of longitudinal measurements in that community. And you could just see that's just one of 10,000 studies that are shared on dbGaP today. Molecular biologists share their data. So this is a project called ImmPort. ENCODE, actually, is a project called ENCODE. ENCODE is the encyclopedia of elements in the DNA. And this project is completed after about 6 or 7 years. You can see by the infographic on the right here, 442 consortium members in 32 different institutes all worked in a cooperative manner with multiple different grants from NIH. And completed almost 1,700 experiments, generating 15 terabytes of data. And all this data is, again, shared with each other and shared with the public. My favorite number on this slide is the bottom right. This team of 400 plus individuals spent 292 person days just on the conference calls to organize themselves. You can see at the bottom, the total cost of just the teleconferencing was 50,000 British pounds. So it's a lot of coordination. No one is saying this is easy, but the point here is that the data, valuable data, is collected and shared. On this slide, you can see that the chemical biologists share their data. What is a chemical biologist? Well, these are the individuals, perhaps, there are many in pharmaceutical companies, but also in academia now, that run drug screening assays. So drug screening is when you get a cell for example, that is glowing when it's living, right? It emits a color. And then you could test 10 or 100 or 1,000 or a million different chemicals or drugs to see if the glowing stops. For example, can you kill the cell, perhaps in a dose-dependent manner. And once it's colorimetric, then you can apply robots to do this kind of screening, so you don't have to do this manually. Of course, this happens in the pharmaceutical companies, but it also happens in academia as well. As part of the NIH Roadmap Initiative, if NIH funded you to do this type of work in academia, you had to have deposited your data into PubChem. And now you can see the numbers at the top right. What is PubChem today? When you just count up all the different rows and columns, think of it as a big matrix, a big grid, where you have 170 million chemicals and drugs and future drugs as the columns, and now already 1.1 million of these screening assays as the rows. If you do that math, that's 190 trillion little boxes, of which 1 billion of them have some measurement of some chemical in some assay, some bioassay or screening assay. That's an amazing amount of measurements, a billion of them out of the 190 trillion that are possible in those combinations. Now, of the 170 million substances, 122 million of them already meet Lipinski's rule of 5. That means they make ideal drugs, perhaps even ideal oral drugs. And a million of the billion measurements are actually deemed active. So this chemical or drug or future drug actually worked. It was active in that screening assay. And this data is not just shared with each other, but this data is given out to the public, even without a user ID and password. An amazing set of data that probably rivals any pharmaceutical company, perhaps even rivals all pharmaceutical companies combined, because this is still growing at an exponential rate. And we give this away to people. And on the next slide, you see another example of data sharing, translational researchers. Now, in this case, these are translational researchers using molecular measurements. On the left there, I'm showing you an Affymetrix gene chip. It's one of my favorite of these kind of assays. And these assays are now ubiquitous in life science research or in the medical enterprise or academic medical centers. And now, because this mandate came from the journals first, for example, the top tier journals, the editors and the reviewers, start to require that raw molecular data be shared with the public. And this mandate really started around 2000, 2001, 2002. And now if you look at these two repositories, one run by the Europeans called Array Express, and one run by the NIH, GEO, Gene Expression Omnibus, you can see 1.7 million of these microarrays publicly available now. That's up from 0 in 2001. And of course, this is growing exponentially. So it's amazing that these chips, which themselves are not inexpensive, they're a couple $100s now, but when run, the mandates are already there, and the community agrees with those mandates to share this data with the public. All of this data is available without a user ID and password. There's no HIPAA sensitivity here. These are de-identified. And moreover, these are RNA measurements, not DNA, so we can't really conceivably, we probably could conceivably, but it's really not practical to re-identify individuals this way. And so this data is downloadable without a user ID and password. Of course, I'm showing you a gene expression microarrays here, but I also mean to cover RNA-seq. I also mean to cover proteomics and mass spectrometry. Of course, these types of data, even when run on patients, are freely shared with the others today. So think about this as the progression, though, that initially the mandates to share data, interestingly, start with the journals. The journals first, then they move to the funding agencies like NIH, Wellcome Trust, Howard Hughes Medical Institute, and then they move to the governments, for example, the FDA or other government mandates. So this progression is one that I've seen multiple times in measurement and data modalities. I think this is the era we're in with clinical trials as well. We start with journals, then we go to funding agencies, and then we go to governments. And so I think we're reaching that mandate in clinical trials as well. On this slide, you see just two of the numerous perspectives and editorials that came out over the past year or two. The one here is in the New England Journal of Medicine. There's multiple in JAMA and others calling for open access to clinical trials data. And the same way, why is it that we can actually share the entire Framingham study and all these molecular measurements, but for some reason we have difficulty doing this with clinical trials data? And I think this is going to change. And I'm going to now, in this introduction, really introduce why do we want to share, what do we get out of sharing, And why are we in this mode where the public is really demanding us to share this kind of data here? So on this next slide, I'm showing you 9 reasons that I found why we are in this mode and why we want to be in this mode of sharing data openly. And I'm going to cover each of these in a little bit of depth. Reproducibility, transparency, supporting public policy, returning data to the community, increasing visibility into failed trials, speeding results reporting, especially showing that to patients, enabling learning and enabling new ventures, and of course, new science, which is actually the fun part from my perspective as a scientist. Let's talk about the first 4 of these. I think here on this slide, I'm just showing the first 4. Now, on the top left, I'm going to talk about reproducibility first. I think it's not really a surprise to perhaps you in the audience that there's this perception that we're in the middle of a reproducibility crisis. And what is this crisis about? Why can't we reproduce experiments in science? I think 2 or 3 years ago, it was noted in one of the Nature journals that a team at Amgen, the pharmaceutical company, the biotech company, tried to reproduce 100 or so experiments, top experiments in the cancer field, and found that only 30% of those experiments actually could reproduce. Now, of course, top scientists say, well, that's what makes the difference between their scientists and Amgen scientists. You could joke about it, but it is a little bit of a concern, of course, to many that why can't the same answers be obtained with the same experimental protocols? Now, if you take this just to the very basics, the easiest way to fix this problem, at least with the most pressing problem reproducibility, is to release one's raw data. In the back there, you can see John Ioannidis' paper in Nature Genetics, which is still, I think, one of the top papers cited in Nature Genetics, on the repeatability of published microarray gene expression analyses. And he showed with these pie charts, you can barely see, that more than half the reasons why they could not repeat the experiment was because they had not shared their data. The authors had not shared their data with the public. It was just unobtainable. And so that is probably the easiest way to at least gain visibility as to why something is or not reproducible, is at least release the raw data so one can see what one did in their experiment. And this is going to be a major driver, I think, for data release. It may not be the most positive driver. We're not building new things with this, but at least this idea of reproducibility may end up getting a lot of data released by those collecting this kind of data. Separate from reproducibility is transparency. So transparency is this idea that we actually want to document better what exactly we do in our processes. And this is getting more important as especially more molecular and translational basic science studies or basic science studies actually are becoming more important and moving closer to the clinic. I think Keith Baggerly's work at the University of Texas MD Anderson Cancer Center against researchers at Duke is the premier example of this, where it took an outside researcher to really call upon this problem that the team at Duke was not being transparent exactly with what they were doing. And this was important because they had launched a clinical trial on their molecular measurements. And as we all know, that this ended up with the halting of the trial and a lot of consternation and a lot of reputations damaged from this type of work. I think we are in this kind of mode where we do not want this to happen again in the field. It is important to get these discoveries to patients, but in a controlled and a transparent manner. So the Institute of Medicine's report on this whole issue, Evolution of Translational Omics, Lessons Learned and the Path Forward, really called for the release of methods and data so that one can see exactly what is going on in a study, especially those that are related to clinical trials. On the bottom right, separate from these, are this idea that data needs to be released to support public policy. I'm just calling one example here is this idea of Tamiflu. And what about Tamiflu? As you know, if you take it within the first 24 hours, Tamiflu may slow the symptoms or perhaps even the dissemination of influenza. Now, this paper, I guess, was originally published in the British Medical Journal, and Roche had committed to releasing this data to the public, but delayed for years, causing a very public fight between the BMJ and Roche on the Tamiflu data. This fight got worse when some of the original authors of that study really could not even recall even seeing some of that data. Why is this important? Because public policy is that if Tamiflu actually works, governments spend hundreds of millions of dollars to stockpile Tamiflu. So taxpayer dollars, serious amounts of taxpayer dollars are actually being used to purchase this drug. And of course it expires, so it needs to be kept up to date. But if taxpayer dollars and public policy is to stockpile this, then that puts an additional burden, one can see, on the release of this data to the public. So one can see exactly why do we believe this. And now, finally, Tamiflu data was released, and it's still not clear to some individuals why, if this drug actually is working or not. So I think as we get to trials that support that are becoming part of public policy, the bar to release that data is going to get even stronger here. Finally, on this slide at the top right is this idea of returning data to the community. Now, what I mean here is not releasing one patient's data, one individual's data to themselves, but their entire self-sustaining patient support communities that are frustrated with the pace of research in their field. So, for example, here, this article in The Scientist that really discusses the Lou Gehrig's Disease Society, ALS, and specifically here, this group wanted to test themselves on whether lithium, the drug lithium, actually alleviates symptoms or not, or improves their outcome or not. And instead, or in addition to trying to get the traditional scientific community to do this, they took this upon themselves as citizen scientists to actually study their own condition. And I guess they found that lithium did not help. Now, the point here is, again, not individual to individual data release, but why not release all the data, all the raw data in a particular field so that other people besides quote-unquote traditional scientists are actually studying these conditions. We have a lot more that could be done by citizen scientists than just a traditional NIH-funded scientist, especially in today's dollar climate and diminishing support from the federal system here. On the next slide, I'm going to show you the next four of these. Visibility into failed trials. I think we all know, especially you clearly know the statistics at a meeting like the Society for Clinical Trials, that probably up to half the trials that are run, 50% of clinical trials, fail at some point. These are including the pharmaceutical company trials like phase 2 and phase 3. And we have this unusual culture, as you all know, that when a trial fails, we often don't even write a paper about it because it's a negative finding. Never mind releasing the data from the trial, we don't even talk about these things. In pharmaceutical companies, it's sometimes even taboo to talk about them. People want to get off those teams as fast as they can. Otherwise, entire programs might be killed. And this has to change. I think, first of all, there's a clear biases that physicians are only seeing the positive answers. They never see the negative trials. And so this creates a large bias in the literature for just in terms of understanding how drugs might work or might not work. But more than that, I think the public could also benefit from seeing exactly why trials do fail. Other scientists could learn why trials fail. Because right now, when data is not released, you could easily consider that the next group that wants to run a similar study does not get any benefit from the previous failure or work that's gone into that field. So visibility into failed trials is a separate motivator here. On the bottom left, I illustrated another example, speeding results reporting. This article came out in October 2013, I think this was in The Guardian, where they announced that a third of large clinical trials had still not published 5 years after finishing recruitment of those patients, after the last patient and patient encounter was involved. This is a big deal because the public had no idea how slow this process was. That after their participation was done, it's still taking up to 5 years to report on those results. If the public really sees how slow the process is, that's going to be a big problem, especially because we obviously depend on them for participation in trials. I wonder how much of this delay or latency is due to inefficiencies, or maybe that could be sped up if one had a concept of data release within some number of years after last enrollment, with an embargo date, so that there's a little bit more pressure to get studies out sooner so that medicine can benefit, patients can benefit. On the top right is another example, another motivator, enabling learning. What do I mean by this? I don't mean just teaching clinical trialists how to analyze data or statisticians how to analyze data. It does occur to me that when you have these large, sophisticated trials, so for example here, I'm imagining trials that have to do with kidney transplantation. Think about a kidney transplantation trial where hundreds, if not thousands, of biopsies are run, drugs are tested, And those biopsies actually are done by state-of-the-art, really top-tier research groups and research labs who are funded and contracted to actually do this kind of work in a core facility. So now imagine thousands of cases of transplant rejection of all sorts of grades and subtleties, strong or high rejection, low rejection, all properly labeled by some of the best teams in the world. And all that data just sits there in a virtual filing cabinet. It does occur to me that that kind of data could be an amazing source of samples and labels to teach the next generation of pathologists. If pathologists can see 1,000 such samples and learn the subtleties from the top labs, from the community, that data could immediately be repurposed just to enable learning. It has nothing to do with the trial, but just the labeling and the samples. And taken to an extreme is the bottom right, this idea of enabling new ventures. When data is released to the public, entrepreneurs out there find all sorts of creative uses for that data, whether it's just the raw release or actually sophisticated release. At the back there, I mentioned this company called NextBio that took the gene expression microdata from one of the repositories I showed you earlier and really cleaned up the annotations, re-annotated a lot, had a great user interface, and then a company called Illumina, the sequencing company, actually purchased them as part of a bioinformatics platform. So they literally got their start and made it all the way through acquisition just with public data. The UK does this with their clinical trials gateway and with the data from the NHS, primary care data from the NHS. More sophisticated use is this logo of a company called Pathwork Diagnostics. Now, they're out of business now, but the concept here is they took some of that raw microarray data related to cancers, and specifically metastatic cancers of unknown etiology. So you see a metastasis, but you don't know what kind of cancer it is. And so they created this was actually the company came out with the first FDA microarray diagnostic that you could put the RNA from that cancer and actually figure out which of 15 types of cancers was that metastasis from. So you're putting the metastasis on the chip, the RNA from that metastasis on the chip, and then you can see where exactly did this cancer come from. Well, how did they learn the right answer? They learned from the public data. The same repository I keep talking about this, the NCBI GEO, the Gene Expression Omnibus. They took public samples. They created a gene predictor, a machine learning predictor with 1,500 genes. They tested it in prospective trials, and then they released this in this kind of way. So it's amazing. FDA diagnostic, but it came out of public data like this. But of course, my favorite example is new science itself. And new science means asking new questions with that data that's public that the original investigators might not have even thought of asking. So this goes beyond a meta-analysis, because meta-analysis is really asking the same question as the original investigators, perhaps with more data. And here we're asking a different question than the original investigators had. I use this paper as an example. This is a paper came out about 2 years ago in Nature Genetics. And you can see this talks about a new gene called a newly discovered role for this gene called ApoBec, A-P-O-B-E-C. And this particular gene they discovered has a mutation that seems to lead to numerous different cancers. So all the little pie charts you see in the right side of the slide are all different cancers: lung cancer, squamous lung cancer, ovarian cancer. And the pie charts really just indicate how many of those cancers seem to have this gene involved as a mutation. Now, it's a big deal to find a new cancer-causing gene, a gene with a mutation. And this obviously is in a top-tier journal, Nature Genetics. Why do I show you this paper? This paper starts in this amazing way. If you look at the methods, the first line of the methods, you can see is we downloaded data from the TCGA data portal. That, to me, is amazing, that a top-tier paper in a top-tier journal with a brand new gene and its role in cancer was discovered because they started by downloading data. This is the new world. This goes way beyond just meta-analysis and informatics. I see much new science coming from existing data like this. And this is what I'm predicting for clinical trials as well. And so what I'll talk about for the rest of this introduction is this example of how we are disseminating data. I know subsequent speakers will be talking a lot about ImmPort, so I'm not going to spend a lot of time on it. But I'm going to toot our horn for a couple seconds or a couple slides, and then really talk about a new science example of how this kind of new science can happen with clinical trials data. Of course, I represent ImmPort. I'm the principal investigator for ImmPort. I've been in that role for a little bit more than 3 years. ImmPort is the archival and dissemination mechanism for NIAID, for transplant data, immunology data, autoimmunity, and we're actually expanding our role to other types of related types of inflammation and immunity data as well. I'm just showing you here the new beta website, which is a lot easier to use than the previous one, and the URL is at the top here. And to me, the next big open data is in clinical trials data. ImmPort was featured in a publication just a little bit less than a year ago in immunology research. So if you want more details, we wrote this up in a couple-page manuscript, and ImmPort has really grown in terms of utility. We currently have over 100 studies now released to the public, and several hundred individuals download these data sets every month now. A lot of these individuals are in academia, but some are in pharma and biotech as well. So they successfully are able to download data. Data continues to move in and out of our system. Many people are now writing up papers that are featuring ImmPort. These are papers not written by us. There's a paper in Nature Biotech on effective sharing of immunology data, and they featured ImmPort as well as other repositories. We were pleased about that. Why would an investigator want to share their data in ImmPort? I love to give this example here. This is a paper that was published in a journal called Science Translational Medicine. It's a top-tier journal. This came out in October 2013 by the senior author is Catherine Blish, who's at Stanford, and she used something called mass cytometry, which is a next-generation type of flow cytometry. And instead of using fluorescence, you use different rare earth elements that are picked up and quantitated by a mass spectrometer at the end of the flow cytometer. And this is a very novel technique. It turns out not many institutions have this setup. So for many students, actually, for them to analyze this data, the only chance they might even see this data as a graduate student might be through these few data sets on ImmPort, because they may not get a machine like this on their campus during their term as a grad student. But let's talk about the author here for a second, Catherine Blish. You can see here on this figure, you can see the sophisticated type of analysis. They can subsort cells into different categories with different signal intensities. And this turns out is one of the first papers that actually cites the ImmPort accession number in the actual acknowledgments. So at the bottom there you see all data are deposited with the accession number SDY232. Now, I can't say cause or effect here, but what happened is after this data was released, 4 people, already 4 teams, just within the first 2 to 3 weeks downloaded this data. So this was downloaded right away by 4 individuals. And already in the first year, this paper was cited 16 times. So this is actually more than you would predict for this journal, given its impact factor. Actually, if you look now, I think it's already been cited in a subsequent 6 more months more than 30 times. I can't say if this is cause or effect, but clearly it's been published by others that if you make your data open, you do tend to have a higher citation rate. Well, let's talk about other kind of anecdotes for sharing. ImmPort does work. Again, I'm not going to spend that much more time on ImmPort. Just another kind of example of data sharing. Now, another reason you would want to share your data through a repository instead of sharing it yourself is really highlighted in this slide. This is an email that got forwarded to me, and it's a nice email. This is a researcher at the Food and Drug Administration, FDA, who's just a basic biologist, a basic researcher, has nothing to do with approval of a drug, but wanted a data set that was collected by immunologists on various different adjuvants, vaccine adjuvants. And this email went out to a researcher at the Mayo Clinic who's coordinating a large team of investigators studying vaccines and adjuvants. And this is the request, just asking for the raw data. Now, the response from the Mayo was just really humorous to me and also just telling. The response from John at the Mayo Clinic was really said here, and I'll just read it, that if we were to send you this data, we would need to get approval from our IRB and get a legal agreement in place in order to transfer the data, and that could actually take several months between the Mayo Clinic and the FDA. But it occurs to us that almost all the data you're looking for is already on a public database at NIH, and we have no problem with you accessing this data as we described below. And so this, again, is another reason, instead of sending your data to the numerous investigators that might want to reproduce your findings, or perhaps learn some biology, that is the role of a repository, is to help you share that data. The kind of example, the corollary or the analogy to this is the mouse researchers. Very few mouse researchers, when they get a knockout mouse to work, very few want to send all the mice to people who request that. But that is the That is the culture of sharing with the mouse researchers. But instead, they use a repository. They use Jackson Labs or Charles River Labs to actually house those mice, and then those companies, those organizations send them out to researchers that request that kind of. There might be a charge involved, but the data or the mice are obtainable in this kind of way. So you might get more citations if you share your data, and it's actually easier when people request. You can just point them to the right direction. ImmPort was featured in 2 IOM reports last year. I'm just proud of that. One on just finding new uses for drugs, and one on improving the effectiveness of genome science translation. And this just really shows the role, because we're among the very first repositories to really share raw clinical trials data with the public. And I mean the entire trials. I mean, we have good ones. We have some with monoclonal antibodies and some novel therapeutics, but we released both arms, not just the control or comparator arms. And we release this without IRB approval. You just need to sign up for an account and you can download this data, the raw table, straight from a contract research organization. And finally, just complete tooting our horn, we've attracted attention of the March of Dimes, a major funder for prematurity research in the United States around the world, as well as the Bill and Melinda Gates Foundation. We're working with both of these institutions now to share their clinical trials data, specifically with Bill and Melinda Gates Foundation, their vaccine data, as well as adjuvant data and those types of immunology studies as well. So enough about tooting our horn. Let's just complete this introduction with a positive example here. And a positive example here I give is really research. It's preliminary research. We've submitted this for publication, and we're waiting to hear back to see what's going to happen with our manuscript. But I'm at least want to talk about this earlier to just give you an example of new types of science from this type of data. This work done by Mazen Nasrallah, who was in my lab, who's now a medical resident at Cleveland Clinic. And he reanalyzed this trial called RAVE. This was a publication in 2010 in the New England Journal of Medicine, comparing rituximab versus cyclophosphamide for ANCA-associated vasculitis. Vasculitis is inflammation of the blood vessels that go, generally blood vessels, and here we're talking about blood vessels to the brain. So this could lead to a stroke. This is a medical emergency and is treated with sophisticated medications, including high doses of steroids. This particular study was powered enough to be randomized. It was a double-blind, double-dummy, active-controlled, non-inferiority study. That sounds sophisticated, and you can see the study design here, where the investigational arm is at the top and standard arms at the bottom, and you can see cyclophosphamide and rituximab, and the 2 different uses. So both groups got cyclophosphamide placebo or cyclophosphamide, and rituximab went into the placebo group there. Now, this is a great study design. You can see all the different types of data that this study collected. And again, most studies are like this: vital signs, heights and weights, the questionnaires, the activities of daily living, of course, blood counts and hematology, chemistry. This study was a little bit more sophisticated because it's a little bit more molecular, and they also had flow cytometry data, as well as obviously the serum ANCA levels. So you have here the various different visit encounter dates. The flow cytometry, repeated serial flow cytometry was obtained, as well as the ANCA measurements. And when we dive into the flow cytometry, you can see all the different panels of flow cytometry that was run. We have one panel that's been released so far on B cells, but you can see that this group also ran T cells and NKT cells. Very sophisticated flow cytometry experiment in this study. Now, first of all this data is released to the public. You can download this entire study, New England Journal of Medicine study, from ImmPort, including this panel of flow cytometry data. But when you actually look right now, even right now, that's 1,100 flow cytometry files. So the first thing we realized is that just because you release data to the public, it doesn't make it easy for the public to actually reanalyze that. In fact, that's a barrier for the public. And by public, I even mean other immunology investigators are busy with their own experiments. Why would they ever look at our 1,100 flow cytometry files to reanalyze this one? So it turns out you need some tools to do this type of reanalysis, or at least help enable the reanalysis. And so on our particular website, we have a tool called ImmPort. Flock. Flock is a flow cytometry clustering type of program, where flow cytometry is cell by cell, looking at the cell surface fluorescence. And what Flock does is actually just to put these into groups, so you can really automate the identification of different groups, whether it's T cells or B cells or novel groups you don't know about. The idea is to use automated tools to gate these flow cytometry files. And And then that way it's a lot faster than doing this one by one. Now, first, remember that when we download a data set now, what's the first thing we think is going to happen is reproducibility. So here's just a very clear-cut example of how to reproduce the findings from this New England Journal of Medicine paper. On the left is the actual figure from the original publications. This is just showing that if you give rituximab, the B cells drop. We, of course, know this. And on the left, you can see days after randomization and the B cell count, depending on the different panels, different arms that the patients were on. And on the right, you see the same exact figure downloaded or created from the data that's been released to the public. And of course, we added colors. We made the tick marks to match the same, so you can see the exact kind of the drop. So this just really shows that you can reproduce the data. The data is released. You can make the same type of figure. Now, of course, just to be really clear here, the original files, flow cytometry files, were manually gated, and these happen to be automatically gated. So we get reasonably good results even with the automation. We don't have to look at all 1,000 files ourselves, and it's enough to reproduce this figure. Now, reproducibility is a great example, great motivator, but it's not a fully positive one. And here's the positive one I want to really leave you all with. Now, if you look at this RAVE study, New England Journal of Medicine, great journal article, great journal. Yet they were not powered enough to say rituximab was better than cyclophosphamide. Now, cyclophosphamide has a number of side effects, short-term side effects. It's an inexpensive drug, and it seems to work 50-50, or specifically 53% success, 47% not success. Rituximab is a new drug. Very few short-term side effects, perhaps some long-term side effects. We don't know yet. And it's a very expensive drug. Yet in this non-inferiority study, all the authors could really say is that this drug works 60-40, or specifically 64% reached the primary endpoint, 36% did not. And technically, that's not statistically significantly better. It's an 11-point difference, but it met the criteria only for non-inferiority. Now, I pretend I am a new rheumatologist. I'm seeing a patient with vasculitis. Which drug do I put them on? I have 50-50 on one side and 60-40 on the other, but I have a cost differential here. And I have a difference in side effects, perhaps short-term side effects, perhaps long-term side effects. So when we look at this, we realize that rheumatologist is still stuck. But one of the things we can do, really as a neutral third party here, looking at this data, we can come in and just ask, do any of the other measured factors predict this response? In other words, is there what we would call a companion diagnostic that we can now, in hindsight, develop for rituximab? Did any marker that was measured really predict which arm to put a patient on? And indeed, when you do this analysis, Mazen found that the granularity cells, the neutrophils, the granular cells, actually were the ones that actually could be used to tell the difference. So those rituximab-treated patients that went into remission, had a higher neutrophil count or granular cell count. Okay, counterintuitive because we think it's B cells, but these are actually granular cells. In other words, if you have a vasculitis patient that has a high granularity count, strongly consider the use of rituximab. If they have a low granular cell count, strongly consider the conventional cyclophosphamide. So the idea here is instead of ending with 50-50 and 60-40, if one were to design a new trial with that as a marker, one could imagine getting up to the 60s, 70s, and even 80% remission rates because you're accurately using precision medicine, right? This is a precision medicine marker to put the patient into the right arm to then run the next study. And of course, if this pans out in the next study, this would be a companion diagnostic for this particular drug and this indication. Now, this kind of work is being done by pharmaceutical companies and biotechs, including Genentech and others. But to our opinion, this is the first time we as a third party, we were not involved with this trial, could actually come up with this marker. Of course, it needs to be validated. I'll be the first to admit it has to be validated in subsequent studies. But I think we're going to see more of these types of approaches of precision medicine or subsetting with trial data that's released to the public. Maybe in a future world, we won't just have one vasculitis study, but we might have 10 vasculitis studies that are published and publicly available. And we can discover these types of biomarkers or companion diagnostics and in one data set and maybe test them in another data set. And then we gain even more confidence to launch them into a trial. So to end this introduction, I want to leave some take-home points. We have great speakers following me here. Data repositories like ImmPort and GenBank and Geo and all the rest are not the end of the line for data. It's the beginning of a new line for data. And I think we're going to enter this new world of researchers, of community researchers that just reanalyze other data to create new science, not just because they're picking on original researchers or picking on modalities or methods, but really to find new discoveries within that data. Many successful people upload and download data into our repository, including clinical trials data, so there's very few excuses now for actually saying that it can't be done. I hope I've convinced you looking at data is good, especially for reproducibility, but reanalyzing it is great. We can reproduce the clinical trials results, we can expand them, we might even discover new markers for personalized medicine or precision medicine. And I really believe that while individual studies are good, I think pooling studies is going to be great. And I think especially pooling across the same types of studies, people know this kind of concept of meta-analysis. I think we're going to learn why certain trials work and fail in particular areas, and we're going to learn some new biology while we do this. And in the end, linking the molecular and clinical trials data, especially as they're getting released, whether it's flow cytometry or other types of molecular data, are going to enable even more virtual findings and studies. So with that, I'm going to close this out, its introduction, with just mentioning the team, a large, long list of individuals on the team here, many of whom are actually in the audience or in the speaker set today. And thank you very much for your patience in dealing with my virtual presentation. Thank you.